Hi everyone, welcome back to the course of BIOL3202 Nutritional Biochem. Today we are going to talk about the first B vitamin called thiamine, and on the screen you'll see its structure. So on the screen you'll see all your learning outcomes. So at the end of this lecture you should be able to understand the processes of absorption, metabolism and excretion of thiamine. Describe the signs and stages of deficiency and toxicity of thiamine. Acknowledge the typical methods for assessing the biochemical status of thiamine. And describe factors affecting thiamine availability for absorption. Now on the screen, you will see the pathway related to energy production. So on this part, you'll see glycolysis. And then on the bottom part, you see your TCA cycle. The reason why this particular pathway diagram is shown here when we talk about thiamine is because thiamine is a useful cofactor for parts of these pathways. So you'll find thiamine here, here, and here. For now, what I want you to remember is that thiamine is important for energy metabolism. We'll get into further details of these biochemical steps later on. For now, just remember thiamine is important for energy metabolism. Thiamine is actually the first of the B-complex vitamins to be discovered, and therefore in the old days is called vitamin B1. Now notice that the number in vitamin B1 is written as a subscript, which is a very important feature of how you write the numbering system of vitamin Bs. But due to the large numbers of B group vitamins subsequently discovered since thiamine, nowadays we commonly refer to the B vitamins by their names instead of the numbers. So while you might still see on the internet or on textbooks vitamin B1, B2, B3, etc., most often you will find the exact name of the vitamin being displayed on textbooks, on supplement labels, etc. And in that way, actually, it is more informative than just giving a number to a vitamin B, because it is very meaningless to show vitamin B1, 2, 3, 4, 5. But if you have the name, at least it gives them some sort of identity. Thiamine, due to its role in energy metabolism, it is indeed very widely available in the food supply, because think about what thiamine does in any living cells. It is an important cofactor for energy metabolism. That means any living organism requiring energy metabolism would have thiamine inside its cells. Therefore, in our food supply, which most of them are made from living organisms, such as muscle meat from an animal, legumes and grains from plants, they all would contain a reasonable amount of thiamine at the very least, if not a lot of thiamine. And depending on the part of the organism that you're looking at, if it is something like meat, for example, which is a part of the body that uses a lot of energy, of course you would expect to see a lot of thiamine contained inside the cells in meat, because thiamine would need to be in abundant supply to support the energy requirement of muscles. Same goes for legumes and grains, which are the embryo of a plant, and in the germination process a lot of energy is required. So the plant has prepared the offspring basically with a lot of thiamine to support the germination. So therefore these parts of the living organisms tend to be the best sources of thiamine in our food supply. Now we'll go through the digestion, absorption, transport and storage of thiamine in further details in the subsequent slides, but basically we'll talk about what we call the anti-thiamine factors, such as thiaminase. We'll talk more about the absorption, but mostly it is in the jejunum by simple diffusion when the intake is high or just at normal dietary doses. Or it can also be absorbed through the help of transporters when the doses is low. Ethanol is something that will interfere with the absorption of thiamine, which will lead to some severe consequences, which we'll talk about. Inside the body, thiamine is transported as free thiamine or bound to an albumin protein, or as what we call thiamine monophosphate, TMD. So, in the food supply, thiamine mostly exists in the form of TPP or TDP, they're the same. TPP stands for thiamine pyrophosphate, which is the active form of thiamine. And therefore, this is the form you normally find in any living cells, because that's the form they use to support energy metabolism. But this form is not directly absorbable, so it has to first be cleaved to form the free form of thiamine. Sometimes you also find the free form of thiamine in the food supply. Now, when the concentration of thiamine is high, okay, when it is high, absorption is through simple diffusion. Doesn't require the use of transporters when the concentration is high, because the concentration gradient is large enough for thiamine to diffuse inside the enterocytes. Whereas when thiamine concentration is low, thiamine is absorbed through the help of two transporters, thiamine transporter 1 and thiamine transporter 2, which will pump thiamine from the low concentration in the lumen inside the enterocyte. And once inside, the thiamine inside the enterocyte would be further pumped out from the enterocyte into the blood circulation. So this is how thiamine absorption is like in general. Thiamine is so important for energy metabolism. Basically, that means if we don't consume enough of it, we will die, because we won't be able to produce energy to support our daily activity. So how much do we actually need? To your surprise, thiamine is something that we actually don't need to consume a lot. We only need to consume it in the very low milligram region. For men, it's around 1 milligram, and for female, it is around 0.9 milligrams. It is very important to note that thiamine requirement is proportional to energy requirement. Therefore, you see that children who are smaller in size and thus having lower energy requirement requires less thiamine compared with adults. And same goes for female versus male, because females tend to require a bit less energy than males, with the exception of pregnancy and lactation, both of which would increase the energy requirement for the women. So where do you find thiamine in our food supply? Tissues with active energy metabolism tend to have more thiamine, as we said. Muscle meat would have the most thiamine, followed by anything that has a lot of energy active cells, such as soy milk, which is made from legumes. And fortified food is another rich source of thiamine in our food supply. Interestingly, when you compare milk with soy milk, milk doesn't contain as much thiamine as soy milk, because think about what they're composed of. Soy milk is made from legumes, which are a very rich source of thiamine because they're cells, whereas milk is not made from any cells. Milk is a food for the young calves to support its growth. So there is no living cells inside milk, but it does still contain some thiamine to support the growth of the living calf. So when compared with soy milk, which is made from living cells, of course, the thiamine content of milk is going to be lower. Thiamine is actually something that's quite stable at low pH, but it will start to decompose when you heat it up, particularly when the condition is non-acidic. So for example, when you cook meat using convection cooking, just such as baking or grilling, you can lose as much as 85% of the thiamine contained in it. If you cook vegetables with water, you can also lose from anywhere between 0 to 60% of thiamine. Pasteurization would lead to about 10 to 20% loss. Thiamine is something that is very sensitive to heat sensitive. But is that going to be a problem? Not really, because thiamine is so abundant in our food supply. We don't really care too much about losing thiamine in our cooking. Look at pork chop here. So this pork chop is about 100 grams, which is about the size of a normal piece of pork chop. And this is cooked pork chop. So look at the thiamine content after the cooking loss. It is supplying close to 80 or 90% of the RDA of thiamine. This is not likely to be a concern because thiamine is so abundant and we eat a wide variety of foods containing thiamine in our food supply. It is fair to say that if you're eating anything in a day, it is extremely unlikely that you will develop thiamine deficiency. Now, this is the structure of thiamine, and you are not expected to be able to draw this molecule in the exam or midterm quiz. What you should remember is that, number one, there is a reactive carbon on the thiazole part of thiamine. Number two, it has a pyrimidine ring, and the two parts of it are joined together by a methylene bridge. So these are the three things that you need to remember, because these are the things that make thiamine active. Without any one of these, this is not thiamine, regardless of how similar the structure is. If you add phosphate groups at this part of the thiamine molecule, you get the active form of coenzyme called thiamine diphosphate, TDP, or thiamine pyrophosphate, TPP. They are the same. Now, thiamine has a cousin that has a very similar structure called thiochrome. It has something that looks like the pyrimidine ring, but basically this part of it is joined to this part to form another ring in the middle. So even though it has something that looks like the thiazole, something that looks like the pyrimidine, it has no methylene bridge, and therefore it is biologically inactive. So you have to have all three together to form the active coenzyme by adding two phosphate groups at this part of the molecule. So to get the TPP, if you have the free form of thiamine, you need to add two phosphate groups to it, and the phosphate group comes from ATP. Now with the help of thiamine pyrophosphokinases, so you get TPP, which is the most abundant form of thiamine inside the human body. The enzyme name is very simple to remember. Kinase is something that adds phosphate to any molecules, and the name of the product molecule is called thiamine pyrophosphate. So it's thiamine pyrophosphokinase. So simple. Another form of thiamine that you will find in human body is the TTP form, triphosphate, three phosphate groups, one more, and it accounts for about 10% of the thiamine that you find in human body. So you add another phosphate group to TPP with the help of ATP, of course, and an enzyme called TDP-ATP phosphorotransferase. So it transfers a phosphate group from ATP to TDP. And to get TDP back from TTP, you need to strip off one phosphate molecule from the TTP. And to do this, you use phosphatase to strip off phosphates. And the enzyme involved is called thiamine triphosphatase because it strips off a phosphate from the triphosphate. These are the different forms of thiamine that you will find in human body. TTP accounts for 10% around, TPP accounts for more than 80%, and then the rest would be the free form thiamine or the TMP form. The main function of thiamine is that it acts as a coenzyme in energy metabolism in two steps of the energy metabolism pathway, the pyruvate dehydrogenase step and the alpha ketoglutarate dehydrogenase step. A brief recap of what coenzyme is. Coenzyme is something that will bind to what we call an apoenzyme. So apoenzyme is an enzyme that is inactive. because the shape of the binding site is incomplete without the coenzyme. Now once you add cofactor or coenzyme onto the apoenzyme, you will form the correct binding site of the enzyme, and this enzyme with the cofactor now attached to it is now called the holoenzyme. So the holoenzyme is the form that has the correct binding site for the substrate. TDP is a coenzyme, and therefore you will now see the importance of the structure of TDP. Without the correct structure, once you bind to the apoenzyme, number one, it might not bind correctly. Number two, even if it binds, the shape here might not be correct, such that the binding site is not going to be in the correct form. If thiochrome is bound to the apoenzyme, for example, you might get thiochrome might look like this. It might still bind properly to the apoenzyme, but the binding site is in the incorrect shape. So it doesn't have the activity that thiamine has. So it has to have the exact structure that I've shown you. The thiazole joins to a pyrimidine ring through a methylene bridge, which is very important. TDP could be found in two steps in the aerobic respiration pathway, the pyruvate to acetyl-CoA conversion, and then alpha-ketoglutarate to succinyl-CoA conversion. There's an additional step in the glucose metabolism pathway, which is the hexomonophosphate shunt, which will also use TDP, which is the transketolase steps, which we'll talk about later. TDP, thiamine, participates in energy metabolism by assisting the conversion of pyruvate into acetyl-CoA. The first step of this is when the reactive carbon on the thiamine molecule attacks the middle carbon of pyruvate to form an adduct, which will then have a carbon dioxide molecule stripped off with the help of pyruvate decarboxylase to form a molecule called hydroxyethyl TDP. This hydroxyethyl TDP would have the hydroxyethyl group, which could be transferred to a molecule called oxidized lipoamide to form acetyl lipoamide. In the process, it will regenerate the TDP. This acetyl lipoamide, as the name suggests, contains an acetyl group, and as you can recall, acetyl-CoA is what is fed into the Krebs cycle. This acetyl group on the acetyl lipoamide will react with CoA to form acetyl-CoA. This is fed to the Krebs cycle. Now, the reduced lipoamide that is formed by the reaction of acetyl lipoamide with CoA will then be oxidized by FAD and NAD system to reform the oxidized lipoamide to start the process again. So you will see that TDP, thiamine, is kind of regenerated rather efficiently in this process. It can be pretty much regenerated many times. So this is how thiamine participates in energy metabolism in the pyruvate dehydrogenase step. Thiamine can also act as a coenzyme in the catabolism of branch-chain amino acids such as isoleucine, valine, and leucine. The step involved is the step that uses the enzyme called branch-chain alpha-keto acid dehydrogenase, or BCKDH. All three BCAA uses the same enzyme, BCKDH, and you see that it requires thiamine as a cofactor. And lastly, thiamine can also participate in the synthesis of pentose and NADPH in the hexomonophosphate shunt in two of the steps involving the use of transketolase. These are the two steps, transketolase, and these two steps require TDP. These steps generate the 5 carbon sugars for nucleic acid synthesis or glyceraldehyde 3-phosphate that could form advanced glycation end product. In terms of metabolism and excretion, thiamine is excreted as an intact molecule or as one of these metabolites. So you see thiochrome here can also be excreted as TPP, TMP, etc. So many different forms. Measuring urinary thiamine is not going to be a very accurate indicator of thiamine status in the body. In terms of deficiency, there is a specific condition that is due to thiamine deficiency called beriberi. There are 3 different forms of beriberi: dry, wet, and acute. And the more severe forms of thiamine deficiency can lead to Wernicke-Korsakoff syndrome. Beriberi, beri means weakness. So beriberi is a condition where the person suffering from it would feel weakness due to the inability to generate energy properly. The dry form of beriberi would come with muscle weaknesses and wasting, especially in the lower extremities, and peripheral neuropathy. The wet form of beriberi would come with cardiomegaly, that is heart enlargement, and tachycardia, so extremely quick heartbeat rates. Because energy production is impaired, so the body senses that it is not getting enough energy from aerobic energy metabolism, so it might think that oxygen supply is insufficient, so it pumps more oxygen around. And then the acute form of beriberi would come with anorexia, vomiting, and lactic acidosis. Why would you get lactic acidosis from acute beriberi? That's because of this part. So to get pyruvate into the Krebs cycle, you need thiamine. Now, you do not have thiamine. This enzyme is not working. You have a lot of pyruvate from glycolysis, and it has nowhere to go, and you need energy. The body would make use of this pyruvate to produce some energy through the conversion of it to lactate. And eventually you have a buildup of lactate, causing lactic acidosis. Thiamine deficiency often occurs in alcoholics, but also it has been identified in non-alcoholics and babies. The classic symptom of alcoholics suffering from thiamine deficiency is called the Wernicke's encephalopathy, which is the first part of Wernicke-Korsakoff syndrome. It will come with constant involuntary eyeball movement, and then ataxia, amnesia, and confusion. All these are reversible symptoms if treated early enough and properly. This is Wernicke encephalopathy on the medical imaging. In panel A, you see someone who has the Wernicke's encephalopathy due to thiamine deficiency. B is the normal brain. And then C is the same person in A after receiving thiamine treatment. So you see that the white part, which represents some sort of damage or lesions in the brain, disappears once thiamine status came back to normal. It could revert back to as close to the normal brain after thiamine supplementation. That's why we said this is completely reversible. No long-lasting consequences if treated early enough. However, there are 2 parts to the Wernicke-Korsakoff syndrome. If the Wernicke's encephalopathy is not treated early enough, then Korsakoff syndrome will follow, or we call it Korsakoff psychosis. Wernicke's symptoms will go away if you don't treat it for an extended period of time. You might mistaken it as having recovered, but indeed it is turning into something worse, which is the Korsakoff syndrome. Wernicke encephalopathy causes brain damage in the lower parts of the brain, called the thalamus and hypothalamus. The Korsakoff psychosis is different from the Wernicke encephalopathy in that it induces permanent damage to areas of the brain that involves with memory. Thiamine often doesn't improve loss of memory and intellect that occur with Korsakoff psychosis. If you see someone with thiamine deficiency with Wernicke encephalopathy, you should indeed try your extra best to revert the Wernicke encephalopathy as quickly as possible, because once it progresses from Wernicke's encephalopathy to Korsakoff syndrome, there is no way back. The damage will become permanent. There's another condition that is very rare but related to thiamine deficiency called the Machiafafa-Bignami disease, which is characterized by the demyelination of the corpus callosum. And this is seen most often, again, in patients with chronic alcoholism. This relates to inadequate availability of thiamine pyrophosphate as the cofactor for energy metabolism, which will reduce the ATP production. And also, it affects the production of acetylcholine, glutamate, aspartate, GABA, and dopamine, all of which are important neurotransmitters. These, together with the effect of ethanol, contributes to the damage to the corpus callosum. Alcohol is really bad for most of the nutrients, and thiamine, unfortunately, is not found in alcoholic beverages. Someone who drinks a lot of alcohol, which interferes with absorption, plus it's not available in the alcohol, plus these heavy drinkers tend not to eat very well. The consequence is, of course, thiamine deficiency. Very logical. You have reduced absorption of thiamine. Alcohol does that. It also leads to poorer dietary intake. Vomiting, diarrhea, steatorrhea would result from chronic alcohol consumption as well. So basically, the intake of thiamine is reduced a lot. Absorption is reduced, and then there is increased demand to metabolize the ethanol. All these together contributes to thiamine deficiency in chronic alcoholics. Now, this shows you the effect of ethanol on the absorption of thiamine. So they measure the thiamine absorption by using radioactive thiamine at a very high dose, 5 times the RDA. So you expect to see the absorption would be very, very efficient by simple diffusion at this dose. It is not when ethanol is present. So this curve here represents the absorption of thiamine when there is no ethanol. Very efficient. It peaks at about 10 counts per 100 minutes per mil. Whereas when there's ethanol, the absorption is cut by as much as 60 or 70%. And this is in malnourished alcoholic patients. These are in healthy subjects. The effect is the same. So how do you assess thiamine deficiency? You need to make use of a test called erythrocyte transketolase activity in the blood, because transketolase is an enzyme that requires TDP as a cofactor. Now, you can't use thiamine in urine as a very accurate indicator, as I've mentioned, because thiamine can be excreted in many different forms of metabolites. But in blood, it seems to work okay. Those who are at risk are, of course, the alcoholics, elderly people because they tend to not eat very well, those who have had bariatric surgery, and those who eat a lot of raw fish or drink a lot of tea, which we'll get into further details. The transketolase activity test is done this way. You have the whole blood from the test subjects, and then you add thiamine, you add TDP to the blood. So you measure the activity before you add TDP, and then you measure the activity of transketolase again after you add TDP. If the difference between the two measurements is less than 15%, that means the addition of TDP does not increase the transketolase activity a lot, implying that the transketolase have been getting adequate TDP supply in the blood already. Whereas if the difference between the two measurements is greater than 25%, that means in the initial test when no TDP was added, the transketolase activity isn't at its best because some of it might not have the cofactor available to them. But when you add the TDP, you will then provide those not having the TDP cofactor with a TDP molecule to complete the holoenzyme. You see an increase in the transketolase activity after the addition of thiamine. So basically what you do is that you measure the amount of glyceraldehyde 3-phosphate produced. you add the TDP or TPP to the blood, which will turn the cellulose 5-phosphate and ribose 5-phosphate to cetylheptulose 5-phosphate and glyceraldehyde 3-phosphate. This is what you use to then perform your assay. This is a standard curve, so color change basically, like your spectrophotometry. Factors that affect thiamine bioavailability includes something called thiaminase. So as the name suggests, this is an enzyme. And thiaminase, the name suggests that this is an enzyme that acts on thiamine. What it does is that it breaks down thiamine. What it does is it cleaves the methylene bridge. Without the methylene bridge to join the two moiety, thiamine activity is lost. There are two types of thiaminase that you find in food. Type 1, which are found in fish, shellfish, ferns, and some forms of bacteria. And then type 2, which are found only in bacteria. But they do the same thing. So they cleave the methylene bridge. The reason why those who eat a lot of raw fish tend to be at high risk of thiamine deficiency is because fish contain a considerable amount of thiaminase, depending on the type you eat. So some fish has no thiaminase activity. Some have a lot, like tuna, which are used extensively for raw sashimi in Japan. And clam, which we don't normally eat raw, contains the most. So this is the thiamine destroyed in terms of milligrams per 100 grams of food per hour. Tuna is a particular problem because quite a bit of people, Westerners, Asians, tend to eat tuna raw. Now, because thiaminase is an enzyme, it is destroyed by heat. So if you eat the fish cooked, that's okay. Thiaminase is only a problem when the fish is eaten raw. If you eat tuna cooked thoroughly, you don't need to worry about the thiaminase at all. It's only when you eat the tuna raw in large amounts, then you might need to start to think about your thiamine status, especially when you do it every day. For most of us, this is unlikely to be a concern. Then there are also some other anti-thiamine substances. These are things that would form adducts with thiamine, making them unusable, such as tannins in tea. Tannins are something that likes to form adducts with any other thing inside the same space. And once it forms the adduct, basically that thing, vitamin, mineral, whatever, is lost because you can't get it back. Tannins are something that would like to hold on to anything that it forms adducts with. If you have a lot of tannin in the diet, some of the thiamine would be trapped by the tannins in the form of adducts. And those who have had bariatric surgery might also be at risk of thiamine deficiency. But those who had this form of bariatric surgery called Roux-en-Y gastric bypass would have the most problem. And that is because of the amount of food that could be consumed. It has little to do with digestion and absorption. It has mostly something to do with the amount of food that could be consumed, because in this form of bariatric surgery, the whole stomach is bypassed. Therefore, this person is unable to consume reasonable amount of food. If this person is consuming normal food, not the formulated food, then thiamine intake is likely to be low because the amount of food ingested is low. The other forms would at least have some parts of the stomach being kept in the digestive tract, so the amount of food consumed can still be reasonably high enough to ensure adequate thiamine intake. This is a study that shows basically what I've just said. Most of the thiamine deficiency symptoms seems to come from the Roux-en-Y gastric bypass patients, and it affects females more than males. And there are also something called thiamine analogs. which are compounds with very similar structures to thiamine but have no vitamin B1 activity. So what they do is they compete with thiamine on the binding site of the APOL enzyme. So they fit into the binding site of the APOL enzyme, but the other side of it doesn't have this correct shape to perform the actual thiamine activity. The holoenzyme that is formed from the binding of the APOL enzyme with these thiamine analogs is in the incorrect shape. Now, thiamine toxicity is generally very low because it is water soluble. Excessive thiamine intake are excreted in the urine. It's directly proportional to oral intake. This is the RDA, and anything above are excreted very efficiently. But very high doses enter the system via subcutaneous infusion or intravenous infusion might indeed induce toxicity. We're talking about more than 100 times recommended intake, and through infusion, not dietary sources. In dietary sources, the absorption is through simple diffusion, so that it is saturable. So there's only so much you can absorb. Anything exceeding the saturation of the absorption limit is not going to be absorbed efficiently. And the transporters are not going to pump the thiamine across because the dose is too high. But when you do it intravenously, then you bypass all of these. So it might indeed cause anaphylactic shock, but that's very rare. This shows you the radioactivity of thiamine excreted after intravenous administration and intramuscular administration of 200 mg dose of thiamine. This is the serum level that's dropping since IV infusion, and then this is the amount of thiamine excreted. So you will still be able to excrete most of these IV injected thiamine efficiently. If you do intramuscular infusion, serum level will go up and then it will slowly come down, because it has to go through the muscle to the blood, and then you'll also be able to excrete most of this because this is a water-soluble vitamin. Now, with thiamine in clinical nutrition, we pay a lot of attention to thiamine as part of the management of what we call the refeeding syndrome. Now, refeeding syndrome are, as the name suggests, something that happens when you refeed someone. You refeed someone when this person has been chronically malnourished or who has gone on a prolonged fast. What this thing does to the body is that it reduces the insulin, increases glucagon, and increases cortisol because the body senses that it is in hunger mode. It requires this change in hormones to ensure survival. The change in these hormones will lead to changes in glycogenolysis, gluconeogenesis, and protein catabolism to supply glucose for energy. It would deplete electrolytes, protein, fats, minerals, and vitamins. Now, at the point where you treat this malnutrition, you refeed them with adequate nutrition, the refeeding would induce the secretion of insulin, which will increase protein and glycogen synthesis, increase glucose uptake from the food, increase uptake of phosphorus, magnesium, and potassium, and increase the use of thiamine because now it has more glucose for energy production. This is where thiamine comes in. Now, this person who has been chronically malnourished indeed actually has thiamine deficiency to start with. Over time, with the refeeding, the food supply, so the diet would supply thiamine, but in the beginning, the person's body store of thiamine is low. This person is thiamine deficient. Now you are giving this person food to induce a condition where it requires a lot of use of thiamine when there's no thiamine available. So this is going to cause problems. In clinical nutrition, to prevent refeeding syndrome, what we normally do is to start the refeeding slowly, such that it doesn't induce an excessive use of thiamine. This is a very common mistake that people make in the clinical practice. When they see someone malnourished, without monitoring this person properly, they will start the refeeding too quickly. This is going to kill them somehow. So this is what you need to remember. What the thiamine deficiency does is that it blocks all the enzymes that requires the thiamine. This person having food intake is unable to make use of the food as fuels for energy production, which will then lead to lactic acidosis, as we mentioned, because the pyruvate dehydrogenase doesn't work. So you have an influx of glucose. The body requires energy, but the glucose isn't able to be used for aerobic energy production because pyruvate dehydrogenase doesn't work in the absence of thiamine. The glucose would be fed into the lactic acid production pathway, leading to a buildup of lactic acid. Lactic acidosis in the severe form can kill a person, together with the disruption of the electrolyte balances. So refeeding syndrome is something that has to be monitored very closely in clinical practice. And this is one of the leading causes of death of people who are being treated for malnourishment. So when you study clinical nutrition, this is one of the most important topics that any dietetic school will teach you about. And make sure that you don't make this kind of mistakes. So that's all for thiamine. And next time we will talk about riboflavin.